speakers of the day. I am Dr. Shweta Bavari from Pharmacy Institute, Noida Institute of Engineering and Technology, Greater Noida. In this lecture, I will be covering pathophysiology of sexually transmitted disease, syphilis and AIDS. In the previous lecture, I have already discussed what sexually transmitted disease are. Sexually transmitted disease, as the name suggests, are diseases caused by microorganisms that spread through sexual intercourse. The common symptoms witnessed in sexually transmitted disease include genital ulcers or sores, urethral discharge, vaginal discharge, cervical infection, lower abdominal pain, scrotal swelling and pain, rectal or pharyngeal inflammation in case where the rectal route of sexual intercourse or oral route of sexual intercourse is involved, and the formation of papules that are eruptions on the skin or the mucous membranes. We will begin with the pathophysiology of syphilis first. Syphilis is a systemic bacterial infection caused by spirochete Treponema pallidum. Due to its many protein clinical manifestations, it has been named the great imitator and mimicker. When I use the term protein clinical manifestations, means that syphilis can result in wide array of symptoms. It can mimic any sort of infection because it produces so wide range of the symptoms that can occur and can vary from individual to individual. It can imitate, it can mimic any type of infection. So that is why it has been named great imitator and mimicker. Venereal syphilis is a worldwide disease of only humans with no animal reservoir. So in this case also, the mode of transmission is from human beings to human beings. There is no animal vector in between, no animal carrier. Syphilis is usually transmitted by sexual contact or from mother to infant. These are the two major modes of transmission of syphilis. So when I use the term horizontal transmission during sexual contact from the infected person to non-infected person. This is horizontal transmission. Now when I talk about vertical transmission occurs from a mother to infant while delivering a baby, or while breastfeeding. So this is vertical transmission from mother to child. These are the two major modes of transmission of syphilis. It may also occasionally be transmitted as a blood-borne infection during blood transfusion, sharing of blood products, sharing of needles or parenteral injections. It can also occur by that mode. Now there are various stages of syphilis. The stages of syphilis include first the infection with Treponema pallidum. In this, growth of organisms at site of infection occurs. Then dissemination to various tissues including central nervous system may occur. Then after the infection, when from the site of infection it has disseminated to the bloodstream, now from this it results in what? Primary syphilis. From the site of infection, what occurs is primary syphilis. So primary syphilis is characterized by the formation of chancre at site of infection and regional lymphadenopathy. We'll discuss this in detail. Secondary syphilis, the first stage is infection at the site of infection, then primary syphilis, then occurs secondary syphilis, which is when Treponema pallidum spreads to the blood, it results in disseminated rash, that means rashes all over the body. Generalized lymphadenopathy. In primary syphilis, the lymphadenopathy is basically restricted to the site of infection. In this case, because it is a sexually transmitted disease, so the site of infection is basically genitalia. So lymphadenopathy, swelling of the lymph nodes occur in that particular region only. But now in secondary syphilis, Treponema pallidum has spread in the entire blood circulation and it results in rashes all over the body, and it results in generalized lymphadenopathy, which means formation of the lymph nodes. There are swelling or enlargement of the lymph nodes all over the body. Then occurs latent syphilis, which is dormant stage. Recurrence of secondary syphilis can occur in this stage basically, but it is a dormant stage. So after a dormant stage, there can be two pathways, the two stages. One where there is no further complication, the syphilis is dormant now, okay, there are no complications, but from there the latent syphilis can also result in tertiary syphilis, the more severe form of syphilis, which results in gamma, that is basically granuloma formation, cardiovascular syphilis, that the infection lodges into the cardiovascular system, late neurological complications, because it can enter into the, it can infect the nerves, the CNS also. Now, based upon these stages that we saw, symptoms of syphilis based upon the stage of infection, primary symptoms appear 10 to 90 days. So, when we talk about the primary stage, the symptoms appear within 10 to 90 days, or basically within 21 days of the infection after exposure. The main symptom for primary stage is progress from the formation of less than 2 centimeter chancre. Okay. What is this chancre? Basically a macule. I told about the macule, basically discolored patches on the skin, right? So when we talk about macule, so macula discolored patches on the skin. This macule then progresses to ulcer over 7 days. So this is basically the macule to papule. The papule means an eruption over the mucosal membrane or over the skin. Then this papule progresses to ulcer. So chancre are basically painless ulcerations. Okay, so these painless ulcerations that may appear within 7 days of the infection. These are basically painless, these ulcers, these chancre are painless, less than 2 centimeter in size, they are solitary. Generally a single chancre appears, a single ulcerative sore appears on glands, labia or fourchette. Localized painless adenopathy also occur, means enlargement of the lymph nodes in the particular region where infection occurs. In the secondary stage, symptoms appear 2 weeks to 6 months later after the exposure, can be concurrent with or up to 8 weeks after chancre. Rashes occur which are diffuse all over the body, or they can be on the trunk. Mucous lesions, condylomata lata. Papulosquamous eruptions also occur. There is patchy alopecia, that is hair loss. There are patches, and from those patches there is hair loss. Fever, headaches, generalized painless adenopathy occurs. Generalized adenopathy, now when I am not talking about localized adenopathy that occurs near the genitalia, I am talking about generalized and painless adenopathy, means enlargement of the lymph nodes all over the body. It is generalized. Neurological symptoms, cranial nerve palsy. Neurological symptoms that include cranial nerve palsies, means loss of functions or some degeneration of the cranial nerves, harm to the cranial nerves, loss of functions of the cranial nerves. It is stated that generally in syphilis, the cranial nerve 2, which is optic nerve, and cranial nerve 8, which is vestibulocochlear nerve, is basically affected. So when this optic nerve and vestibulocochlear nerve is affected, this means it is going to result in symptoms where there is impairment of vision, or there will be impairment of hearing, and there will be impairment of the balance, there will be dizziness, vertigo, eye redness or pain. It may result in meningitis if it gains access to the meninges or the subarachnoid space. Changes to the mental status or memory. In latent stage, there are no symptoms because it is a dormant state. The tertiary stage, which I told, the most severe form of stage, where syphilis results to irreversible damage to any organ, because syphilis has moved to the blood, and from there it lodges infection in any organ. So the symptoms basically occur based upon the organ that is affected. Okay, so if the organ affected is liver, the symptoms will occur accordingly, and it basically results in irreversible damage. The damage is primarily neurological, cardiovascular or gametous, that is formation of granulomas, most commonly in the liver. Then pathophysiology of syphilis, how this syphilis causes infection in human body and how the disease progresses, we will see that. The outer membrane of Treponema pallidum does not contain lipopolysaccharide and has relatively few surface exposed transmembrane proteins. So, it is very less antigenic. This paucity of surface exposed pathogen associated molecular patterns or any antigenic proteins enables the spirochete, this Treponema pallidum, to avoid triggering host innate surveillance mechanisms, or it avoids the triggering of the host immune response, facilitating local replication and early dissemination. So, this basically the Treponema pallidum has very limited number of surface proteins. It doesn't exposes any antigens. So, when there are no antigens, there can be no triggering of the immune response in the human body. So, this is the unique characteristic feature of Treponema pallidum, that it has scarcity of the surface antigens over its surface. So, due to this, it escapes the host defense, and this allows its replication and early dissemination. Means as soon as it enters, it lodges the infection, it enters into the bloodstream immediately. That is what dissemination means. Its limited surface antigenicity promotes evasion of the adaptive immune responses, that is no triggering of the T lymphocytes or B lymphocytes and no formation of antibodies to fight Treponema pallidum infection. Collectively, these attributes have earned Treponema pallidum its designation as the stealth pathogen, because it moves like a thief. It cannot be detected by the host immune system. Transmission of venereal syphilis occurs during sexual contact with an actively infected partner. Spirochetes directly penetrate mucous membranes or enter through abrasions in the skin. Now, it is during the sexual activity, spirochetes can directly penetrate the mucous membranes or it can enter through abrasions in the skin. To establish infection, Treponema pallidum must adhere to epithelial cells and extracellular matrix components. So, first step here also is after it enters an uninfected person's body, they first they adhere to the epithelial cells and the extracellular matrix components. Then fibronectin and laminin are key substrates for these interaction. Then once below the epithelium, once they have entered the epithelium, organisms multiply. This Treponema pallidum, it multiplies locally and begin to disseminate through the lymphatics and bloodstream. As I already told, they do not wait for long. As soon as they, from the primary site of the infection, that is generally the genitalia during the sexual intercourse, they immediately enter into the bloodstream or into the lymphatics. Why? Because they don't have much, there is scarcity of the antigenic proteins on their surface. So, they can escape the host immune response, they cannot be detected by the host immune system. Then they disseminate, they enter into the lymphatics and the bloodstream. Penetration of the blood-brain barrier occurring in as many as 40% individuals with untreated early syphilis can cause devastating neurological complications. So, basically in 40% people it is seen that syphilis or Treponema pallidum can basically pave its path to the CNS. In 40% individuals, this have been seen that it can cross the blood-brain barrier and can reach the CNS, resulting in various neurological complications. Following dissemination, pathogen sensing in host is eventually triggered. Now, when it evades, it escapes the host immune response a lot. But after it has spread a lot inside the body, it keeps on multiplying. It reaches blood, it multiplies. It reaches the lymphatics, it multiplies. It reaches CNS or any body organ system, it multiplies. So, this much load of microorganism is somehow not going to go undetected by the host immune system, right? It will be detected individually. So, when it has spread a lot, it becomes detectable. So, eventually the host immune response is triggered. the organisms are taken up by the dendritic cells, which then traffic to draining lymph nodes to present the treponemal antigens to naive B cells and T cells. Now, why the symptom, that is lymphadenopathy, very common with syphilis is because dendritic cells engulf these microorganisms, this treponema pallidum. The work of macrophages and dendritic cells is basically to damage and destroy these microorganisms and release their antigens out. When the antigens are released out, they are presented to the B cells and T cells for further action. This is how the immune system works in our body. So, the dendritic cells, they carry them to the lymph nodes. So, when the lymph nodes, when they will be carried to the lymph nodes, there will be the multiplication of B cells and T lymphocytes. When they will multiply, eventually the enlargement of the lymph nodes also occurs. This is the reason for lymphadenopathy. This triggers immune response and production of tissue damaging cytokines such as tumor necrosis factor and interleukin 6 are produced that result in wide array of syphilitic symptoms. So, eventually when B cells and T cells are triggered, they not only target the microorganisms, the pathogen which is causing syphilis, but they also release mediators like interleukin 6, tumor necrosis factor alpha, which are also damaging to the host cells, and this results in the occurrence of the wide range of the entire repertoire of the symptoms of syphilis. Now, we come to the pathophysiology of AIDS. AIDS, or acquired immunodeficiency syndrome, is basically a symptomatic HIV infection. HIV stands for human immunodeficiency virus. So, when we say AIDS, AIDS is basically symptomatic HIV infection. That is, the HIV infection is not itself a disease. AIDS is basically the outcome of the opportunistic infections that occur due to HIV infection, and that is basically termed AIDS. It is the most advanced stage of HIV infection. Of course, AIDS only occurs when the HIV, in the advanced stages of HIV, where the immune system of the immune cells of the host are extremely compromised, which allow the entry of opportunistic organisms, which allow opportunistic infections to take place. That is when the HIV infection is said to cause AIDS, acquired immune deficiency syndrome. AIDS is a sexually transmitted disease, but it is also transmitted via blood transfusions, sharing intravenous needles, and from mother to child during the birth process and breastfeeding, like we just discussed in syphilis. The human immunodeficiency virus targets the immune system and weakens people's defense against many infections. So, what is the basic target of human immunodeficiency virus? The immune cells in the host body. The hallmark of human immunodeficiency virus infection is gradual loss of CD4 T lymphocytes, CD4 positive helper T cells, and progressive impairment of immunity, followed by the occurrence of opportunistic infections or cancers that leads ultimately to death, or we can say before death, we can say it ultimately leads to acquired immune deficiency syndrome. Okay. So, what are the hallmark of human immunodeficiency virus infection? It basically will result in the killing, the destruction of CD4 positive helper T cells. When excessive destruction of the helper T cells occur, which are very crucial for any immune system and also immune response, when their count reduces, the immune system in the host body is compromised. When immune system is compromised, it allows for other pathogens to invade the body and cause opportunistic infections. They get opportunity to produce infection in the host body. This ultimately leads to acquired immunodeficiency syndrome, which is called AIDS, and this ultimately leads to death. There is no cure for HIV infection. However, with increasing access to effective HIV prevention, diagnosis, treatment and care, including for opportunistic infections, HIV infection has become a manageable chronic health condition, enabling people living with HIV to lead long and healthy lives. This means no cure is available for HIV infection. Once HIV, because HIV infection is permanent, once it infects the cells, it integrates its genetic material in the host cell itself. So, it is permanent. Every time the host cell will divide, every time there will be DNA replication, the genetic material of HIV virus, the human immunodeficiency virus, will also multiply. It is permanent, it is not going anywhere. But this infection can be curtailed, like it has infected say 100 CD4 positive T lymphocytes. It can be curtailed to this number of cells only, and not allowing it to spread to other cells by the drugs that are available in market, by various preventive measures that are available. HIV is a member of the lentivirus family, a subgroup of retrovirus with RNA as genetic material. So, the genetic material of HIV is RNA. HIV infection in humans is caused by two related yet distinct viruses, HIV-1 and HIV-2. Of both the types, HIV-1 is more virulent, means it causes more severe infection, and HIV-2 is typically less virulent as compared to HIV-1. HIV, now we talk about how this infection occurs in human body and how this disease progresses. So, pathophysiology of AIDS is such as HIV is a spherical virus that attaches to the host cells with its glycoproteins, with external glycoprotein and the transmembrane protein. During the entry process, I'll just read this out once, then I have a figure to show. In that I'll explain these all over again. So, during the entry process, glycoprotein 120 attaches to the cell membrane by first binding to CD4 receptor, CD4 positive receptor. The actual fusion event takes place within minutes by pore formation and release the viral coat into the cell cytoplasm. After the coat disassembles, the viral genome is reverse transcribed into DNA by the virus's own reverse transcriptase enzyme. The viral protein then inserts the viral genome into the host chromosomal DNA. Production of viral particles need host-driven as well as viral-driven transcription. Viral proteins are transported to assemble in proximity to cell membrane. Virus egress from the cell and release of mature infectious variants. This is the complete cycle how this infection occurs and spreads, that I am going to explain through this image. Now, focus on the first point, that is attachment. This is a T helper cell, T lymphocyte, T helper cell, which contains CD4 proteins. CD4 acts as receptors for these viruses. Okay, so the glycoprotein 120, which is present on the spikes of the HIV virus, it attaches to the CD4 protein. It requires an attachment to enter inside any cell. So, that attachment is provided by CD4 receptors that are present on T helper cells. Alright. So, the first step is attachment. Second step is fusion. Once it attaches to the CD4 receptor, this allows its fusion with the T helper cell membrane. So, when it fuses with the helper cell, the cell membrane, it results in the third step, which is uncoating. The virus coat is removed and it allows for the entry of the genetic material. The viral coat is removed and the genetic material enters inside the host cell, the T helper cell. So, it begins with CD4 proteins that are present on T lymphocytes. This is very important. So, first step is attachment, second is fusion, third is uncoating and release of the genetic material of the virus, which is RNA. Now it has entered inside the host cell. Now, here what happens now, because the genetic material of host, that is humans, is DNA and the genetic material of HIV is RNA. So, because it wants to integrate with the host genetic material for permanent infection, lifelong infection, therefore it needs to convert this genetic material RNA to DNA. So, for that, virus has its own enzyme, that is reverse transcriptase. So, the next step is reverse transcription that results in the conversion of this RNA to complementary DNA. This complementary DNA, with the help of again the viral enzyme integrates within, it lodges between the host DNA. Now, as many times the host genetic material will multiply, the viral genetic material will also multiply, and during the process of transcription and translation, the viral proteins are also produced, the entire and these viral proteins then assemble to form small viruses which are called virions. These virions are then released from the cell by egress, and these virions, when they are released, they go and infect other CD4 positive T lymphocytes. This is the whole process. So, basically what happens in HIV infection or in AIDS, the CD4 positive T cells are targeted, and this results in their destruction. This results in lot of destruction of the CD4 positive T lymphocytes. Initially, to match this, initially when the number of HIV virus inside the body are less, the CD4 destruction and the synthesis of CD4 positive T cells is in equilibrium. But when on per day basis 100 billion of HIV viruses are produced inside the host body, at that time CD4 T cell count reduces markedly, and this results in destruction of CD4 positive T cells, and host immune response is largely compromised. The symptoms of HIV infection include: in the first few weeks after initial infection, people may experience no symptoms or only influenza-like illness, including fever, headache, rash or sore throat. As the infection progressively weakens the immune system, they can develop other signs and symptoms such as swollen lymph nodes, weight loss, fever, diarrhea and cough. Without treatment, they could also develop severe illness such as tuberculosis. These are opportunistic infections, cryptococcal meningitis, severe bacterial infections, and cancers such as lymphomas and Kaposi's sarcomas. So, these are all the symptoms of HIV infection and ultimately AIDS, acquired immunodeficiency syndrome. This is all. Thank you.